Hello students, welcome to EPG Pathshala. I am Professor Ramesh Kothari from Saurashtra University, Rajkot, Gujarat. Today we are going to discuss about a module on entry of fructose and galactose under paper carbohydrate metabolism. So students, let us see what we are going to learn in this module. We will try to learn about what are the major pathways for fructose metabolism, specialized pathways of fructose metabolism, and metabolism of galactose. Fructose malabsorption, formerly named as dietary fructose intolerance, which is also known as DFI, is a digestive disorder in which absorption of fructose is impaired by deficient fructose carriers in the small intestine's enterocytes. This results in an increased concentration of fructose in the entire intestine. Occurrence in patients identified to be suffering symptoms of irritable bowel syndrome is not higher than occurrence in the normal population. However, due to the similarity in symptoms, patients with fructose malabsorption often fit the profile of those with irritable bowel syndromes. In some cases, fructose malabsorption may be caused by several diseases which cause an intestinal damage, such as celiac disease. Fructose malabsorption is not to be confused with hereditary fructose intolerance, a potentially fatal condition in which the liver enzyme that break up fructose and deficient. Sucrose, a disaccharide, contains glucose and fructose as monomer. Sucrose can be utilized as a major source of energy. Sucrose include sugar beets, sugarcane, sorghum, maple sugar, pineapple, ripe fruits, and honey. Corn syrup is recognized as high fructose corn syrup, which gives the impression that it is very rich in fructose content, but the difference between the fructose content in sucrose and high fructose corn syrup is only 5 to 10%. HFCAS is rich in fructose because the sucrose extracted from the corn syrup is treated with the enzyme that convert some glucose in fructose, which make it more sweet. Fructose is a product of sucrose hydrolysis and is probably the second most abundant dietary sugar after glucose. Following is the major pathway of fructose metabolism. Fructose is phosphorylated by ATP through the action of two different kinases. Yeast glucokinase form fructose 6-phosphate. In liver and muscles, fructose is phosphorylated by a specific fructokinase yielding fructose 1-phosphate. Fructose kinase is found in the liver, kidney, and small intestine. This enzyme has a very low Michaelis constant, that is a high affinity for fructose, and the activity of this is not affected by filling fasting cycle or by insulin. Phosphofructoaldolase, which is also known as aldolase B, catalyze the cleavage of D-fructose 1-phosphate as shown in figure. Phosphofructoaldolase is an isoenzyme of the glycolytic pathway aldolase. It is abundant in the liver. The fate of D-glyceraldehyde may take several routes. It can get phosphorylated and enter into the metabolism by the glycolytic pathway or the gluconeogenic pathway, or it can undergo oxidation to deglycerate by an NAD plus linked glyceraldehyde dehydrogenase and then be converted to L-serine via hydroxypyruvate, or it reduced to glycerol by an NADH plus linked glycerol dehydrogenase followed by phosphorylation to glycerol triphosphate. The latter may be used for gluconeogenesis or triglyceride synthesis. Figure shows the specialized pathway of fructose metabolism. In specialized metabolic pathway of fructose, first I will explain it in spermatozoa. Fructose is the major energy source for spermatozoa and is formed from glucose in the seminal vesicle. The pathway involved reduction of glucose to sorbitol and oxidation of the latter to fructose. The fructose concentration of semen may reach to 10 millimolar. Most of this is available for the spermatozoal because fructose is utilized sparingly by the other tissue that come in contact with the seminal fluid. The mitochondria of sperm are the only such organelles to contain a lactate dehydrogenase. Because this enzyme is present in the lactate that is formed by fructolysis, can be completely oxidized to carbon dioxide and water molecules without the need for a shuttle system to transporters, reducing equivalent into the mitochondria. The following figure shows sorbitol metabolism in diabetic patient. The formation of sorbitol from glucose proceeds rapidly in the lens of the eye and in the Schwann cells of the nervous system. Sorbitol cannot pass through the cell membrane, and in the diabetic, sorbitol level build up in the cells because the rate of oxidation of sorbitol to fructose is decreased. It is thought that the elevated sorbitol concentration causes an increase in osmotic pressure, which might be a causative factor in the development of the lens cataract and the neutral dysfunction that occurs in diabetes. Defect in fructose metabolism. Essential fructosuria, hereditary fructose intolerance, and consumption of high fructose are the normal defects that found in fructose metabolism. The essential fructosuria. In this case, fructose is not converted into fructose 1-phosphate due to the deficiency of enzyme hepatic fructokinase. Restriction of dietary fructose is the remedy. Hereditary fructose intolerance. Hereditary fructose intolerance is a genetic disorder characterized by vomiting and hypoglycemia after ingestion of fructose or fructose-producing foods, that is sucrose, sorbitol, etc. It is an autosomal recessive condition involving a deficiency of fructose 1-phosphate aldolase. Eating a meal containing fructose causes an accumulation of fructose 1-phosphate in tissue, which is believed to underlie the hepatic and renal damage found in the condition. Several enzymatic activities are inhibited by high cellular level of fructose 1-phosphate. That is fructokinase. Its deficiency lead to fructosemia and fructosuria. Hepatic glycogen phosphorylase. The lack is in part responsible for the postprandial hypoglycemia, and glucagon unresponsiveness. Fructose 1,6-diphosphate aldolase, without it gluconeogenesis is blocked, contributing to the postprandial hypoglycemia. The consumption of high fructose. Fructokinase rapidly converts fructose to fructose 1-phosphate. The activity of the enzyme aldolase B is relatively less, and due to this, fructose 1-phosphate accumulate in the cell. This leads to the depletion of intracellular inorganic phosphate level. The phenomenon of binding of pyrophosphate to the organic molecule lead to the less availability of pyrophosphate for essential metabolic function and is known as sequestering of phosphate. Due to the decreased availability of pyrophosphate, which happens in overconsumption of fructose, the liver metabolism is adversely affected. This includes the lower synthesis of TAP from ADP and pyrophosphate. High consumption of fructose over a long period is associated with increased uric acid blood leading to gout. This is due to the excessive breakdown of ADP and AMP to uric acid. Hereditary defect in fructose metabolism are essential fructosuria, fructose intolerance, and consumption of high fructose. In case of essential fructosuria, fructose is not converted to fructose 1-phosphate due to the deficiency of enzyme hepatic fructokinase. Restriction of dietary fructose is the remedy of this deficiency. Now, essential fructosuria. In case of essential fructosuria, fructose is not converted into fructose 1-phosphate due to deficiency of enzyme hepatic fructosuria. Restriction of dietary fructose is the remedy of this deficiency. Fructose intolerance. Hereditary fructose intolerance is a genetic disorder which is characterized by vomiting and hypoglycemia after ingestion of fructose or fructose producing food. It is an autosomal recessive condition as discussed before, and eating a meal containing fructose causes an accumulation of this fructose 1-phosphate in the tissue. Several enzymatic activities are inhibited by high cellular level of fructose 1-phosphate. In this case of fructokinase rapidly converts the fructose to fructose 1-phosphate, the activity of enzyme aldolase B is relatively less, and due to this, fructose 1-phosphate accumulate in the cell. This leads to the depletion of intracellular inorganic phosphate level. The phenomenon of binding of pyrophosphate to the organic molecule lead to the less availability of pyrophosphate for essential metabolic function and is known as sequestering of phosphate. Due to the decreased availability of pyrophosphate, which happen in overconsumption of fructose, the liver metabolism is adversely affected. This includes the lower synthesis of TAP from ADP and pyrophosphate. High consumption of fructose over a long period is associated with increased uric acid blood leading to gout. Now, let's see about galactose. What happens? Galactose utilized in E. coli is an inducible process. The capacity of the constitutive transport system is depressed in a striking way when growth of E. coli occurs in the presence of galactose and certain compound related to galactose. The cell's growth on glucose or lactose show little activity towards galactose, and the level of galactose accumulation is also decreased by growth in the presence of certain galactosidase inducers such as isopropyl thiogalactoside and methyl thiogalactosides. Growth of the organism in the presence of trace of galactose which are not utilized yield cell which have much smaller capacity to accumulate galactose compared with that of cell which have been cultivated on the same carbon sources in the absence of galactose. The reduced capacity for galactose can be shown to cause by an increase in the rate of loss of galactose from the cell rather than by a decrease in the rate of galactose uptake. So students, we know that three inborn errors of galactose metabolism happens. These are, the most important is classic galactosemia due to galactose 1-phosphate uridyl transferase deficiency. A complete or near complete deficiency is life-threatening with multi-organ involvement and long-term complications. Partial deficiency is usually but not always benign. Uridine diphosphate galactose 4 epimerase deficiency exists in at least two forms. The very rare profound deficiency clinically resembles classical galactosemia. The more frequent partial deficiency is usually begin. Galactokinase deficiency is extremely rare and most incident since the result in the formation of nuclear cataracts without provoking symptoms of intolerance. The Fanconi-Bickel syndrome is a congenital disorder of galactose transport due to GLUT2 deficiency leading to hypergalactosemia. Other secondary causes of impaired liver handling of galactose in the neonatal period are congenital port systematic shunting and multiple hepatic artery venous malformations. Metabolism of galactose. Lactose is hydrolyzed to yield galactose and glucose in the small intestine of infant and small children by one form of enzyme, which is enter the blood stream from the intestine. An adult form of the enzyme is less active and is missing in many adults, leading to a low tolerance for milk and for milk product containing lactose. Galactose is phosphorylated by galactokinase to form galactose 1-phosphate. Hexose 1-phosphate uridyl transfer catalyzes the transfer of UDP from UDP glucose to galactose 1-phosphate to form UDP galactose and glucose 1-phosphate. And epimerase convert UDP galactose to UDP glucose. So we can say that regarding the entry of fructose, the metabolism of fructose, which is also known as fructolysis, through the metabolism of glucose through glycolysis, use many of the same enzyme and intermediate structures as those in fructolysis. The two sugars have very different metabolic fate in human metabolism. Unlike glucose, which is metabolized widely in the body, fructose is metabolized almost completely in the liver in human, where it is directed towards a replacement of liver glycogen and triglyceride synthesis. Under 1% of ingested fructose is directly converted to plasma triglyceride. As per report, 29 to 54% of fructose is converted in liver to glucose, and about quarter of fructose is converted to lactose. 15 to 80% is converted to glycogen. Fructose is a dietary monosaccharide present naturally in fruit and vegetables, either as free fructose or as a part of the disaccharide fructose, and as free monosaccharide in honey. It is also present in form of refined sugar, including granulated sugar, that is white crystalline table sugar, brown sugar, confectioner's sugar, and turbinado sugar. Refined crystalline fructose and as high fructose corn syrup. About 10% of the calorie contained in the Western diet are supplied by fructose, that is approximately 55 grams of sugar per day. As reported, unlike glucose, fructose is not an insulin secretagogue and can in fact lower circulating insulin. In addition to liver, fructose is metabolized in intestine, testes, kidney, skeletal muscle, fat tissue, and brain, but it is not transported into cell via insulin sensitive pathways. While in case of galactose, which is the metabolized from the milk, sugar, lactose, a disaccharide of glucose and galactose, enter glycolysis by conversion to glucose 1-phosphate. This occurs through a series of steps that referred to as Leloir pathway, named after Luis Federico Leloir, who determined the overall process of galactose utilization. Galactose can exist in two different stereoisomeric forms, that is alpha-D-galactose and beta-D-galactose. The alpha form is that which is metabolized in the Leloir pathway. Conversion of beta form of galactose to the alpha form require enzyme galactose mutarotase encoded by GALM gene, also known as aldose 1 epimerase. The most common disorder, as shown in figure, of galactose metabolism is galactosemia. This is due to the deficiency of enzyme galactose 1-phosphate uridyl transferase. It is a rare congenital disease in infant and is an autosomal recessive disorder. An excessive accumulation of galactose is due to inborn error of galactose metabolism. Aldol reductase convert excess galactose into galactol. Galactitol is associated with development of cataract. Accumulation of galactose 1-phosphate in liver result in the depletion of inorganic phosphate for other metabolic functions. Symptoms include loss of weight in infant, jaundice, mental retardedness, etc. In several cases, aminoaciduria, albuminuria are also observed. Diet deprived of galactose and lactose is the remedy of this disease. Another disorder is galactokinase deficiency. In this condition, there is an accumulation of galactose in blood and tissue. In this defective enzyme, galactokinase, responsible for the phosphorylation of galactose, will also result in galactosemia and galactosuria. Here again, galactose is shunted to the formation of galactitol. Another disorder is a deficiency of uridyl diphosphate galactose 4 epimerase. This is autosomal recessive disorder. This deficiency prevents the conversion of modified form UDP galactose to another modified form, that is UDP glucose. As a result, compound associated with galactose processing gets accumulated to toxic level. Symptoms of this include cataract, intellectual disability, kidney, brain, and liver are damaged. So students, let us summarize what we have learned in this module. We know that sucrose and high fructose both supply a significant portion of the total calories in the diet. Sucrose and fructose consist of two simple sugar, that is glucose and fructose. Glucose also comes from starch like potatoes. Our body produces it, and every cell on the face of the earth is glucose in it. Glucose is a molecule absolutely vital to life. Fructose, however, is not. Humans don't produce fructose, and throughout evolutionary history have never consumed it except seasonally when fruit were ripe. Glucose and fructose are metabolized very differently by the body. It is important to note that while every cell in the body can use glucose, the liver is the only organ that can metabolize fructose in a significant amount. When people eat a diet that is high in calorie and high in fructose, the liver gets overloaded and start turning the fructose into fat. Lustig and other scientists believe that excess fructose consumption may be a key driver of many of the most serious diseases of today, including obesity, type 2 diabetes, heart diseases, and even cancer. So, this is all about entry of fructose and galactose. Thank you.